Finish with our session. So thank you again to all the speakers of the today. So it was brilliant. So thank you so much again. So now we have time for the brief talk session. So as yesterday, just few information. So all the brief talks have been pre-recorded. And they will last 10 minutes each, and then we'll have 5 minutes for each brief talk.
So I'm going to introduce our first speaker, Trinh Nguyen from the University of Vienna. And today she's going to present her talk titled The Role of Proximity and Touch for Neural Synchrony in Mother-Infant Dyads. Wait a sec. Okay, perfect.
Hi all, welcome to my talk on proximity and touch and how it's associated with neural synchrony and naturalistic mother-child interactions. My name is Trinh Nguyen. I'm a PhD student at the University of Vienna working together with Stefan Hild. And I'm very happy to be able to present our results with you today. So first of all, I want to thank all the families who participated in the study. I want to thank the lab that I'm working in, Stefan Hild's Wiener Kinderstudien at the University of Vienna, all the research assistants, medical technical assistants who helped us making this project even possible, and of course our collaborators, Bennett Bertenthal and Drew Abney and Dina Salamander. Social interactions are essential to us as human beings. They are even more fundamental to an infant who's just coming into the world, having limited abilities in communication, and especially verbal communication, of telling others how they feel, what they need, and that this early exchange of information is actually essential to building the first bridges of the infant to the world. This can help infants affiliate with others. This can help infants to start to understand others, and by understanding others, also understanding themselves. What we know this far is that there are different modes of communication, and in the non-verbal sphere, touch and proximity are actually one of the most fundamental ones. Interestingly, it's less researched empirically than its importance is stated in the theory. So this is something I've been interested for a very long time, and I'm very happy that we're actually be able to dive into this further in this current study. Monterosa and McGlone have stated in 2020 that touch and proximity are also tools to help the dyad co-regulate, so help the dyad bridge and repair miscoordination during an interaction, which are totally normal and functional, to then reach a state of flow and harmony again, which is also described interpersonal synchrony. So while there's much empirical evidence for behavioral synchronization, there's actually a very little or small body of research on neural synchronization in early development. Starting in 2017 with Victoria Leong and colleagues' study on how mutual gaze affected neural synchronization between infants and experimenters, there has been now quite a few studies, and they all show that the adult or the parent in some cases and children synchronize their brain activity during social interactions. This is actually related to coordination and communication. So the more effective or the more successful the coordination or the communication is, the higher also neural synchrony is in those cases. It's, as I said in the beginning, potentially facilitated by rhythmic behaviors. So Leong et al. Have found its relation to mutual gaze, and there have been studies showing its relation to positive affect. But we know very little about the role of touch and proximity in these cases, even though there has been evidence in the adult literature. What is more is that we also still don't really know much about the function of neural synchrony to child development, to the dyad, both short-term and long-term. So in the current study, we're not going to solve the whole puzzle just yet, but we're just going to look at the first question that I posed this far. So what kind of behaviors feed into neural synchronization between mother and child, and under which conditions, because social interaction is a very broad construct, as we all know. So here we'd like to focus on touch and proximity and mother-infant interactions between 4 to 6 months. So at a very early stage where the interaction is still very much dyadic and less object-focused. We invited 81 mother-infant dyads to the labs. Infants were 4 to 6 months of age, and we measured both dual FNIRS, so dual functional near-infrared spectroscopy, to assess brain activity, and we measured dual ECG to assess respiratory sinus arrhythmia, which is an index for vagal activity. I'm not going to touch upon these analyses here, but you're happy to read them in our most current published paper in Neuroimage. And then, of course, we had video recordings from which we then coded behavior such as touch and divided or categorized that touch into different forms of touch as well. So mother and infant engaged in 3 different kinds of conditions. The first 2 are the distal joint watching and the proximal joint watching condition. Mother and infant were looking at a screen which showed a calm aquarium video. In the distal joint watching condition, the infant was seated next to the mother in an infant high chair, while in the proximal joint watching condition, we changed the proximity, the physical proximity of the infants to the mothers. Therefore, the infant sat on the mother's lap. Then next, we let everything loose, more or less, and let them engage in a free play condition for 5 minutes. So as for the functional near-infrared spectroscopy, which is a non-direct measure of brain activity, because it measures the oxygenation changes, level changes in the blood. What we did here is that we had 22 channels over the inferior frontal gyrus and the prefrontal cortex, all frontal regions associated with important social processes, social cognitive processes. And we took the 2 time series of mothers and infants, or the 22 in total, and related them to one another using wavelet transform coherence, which considers the frequency band they synchronize in, and that the synchronization can be in phase, anti-phase, and phase lagged to a certain degree. So let's dive into the results. What we first looked into is just the neural synchronization of mother and infant on the y-axis in the different conditions on the x-axis. So DW is distal watching, proximal watching, FP free play in the different brain regions. So IFG, LPFC, MPFC, as you can see in the facets or in the columns. What we can see first is that in the inferior frontal gyrus, there were no significant differences between the conditions in neural synchronization, while in both LPFC and MPFC, the free play condition showed the highest amount of neural synchronization between mother and infant. So this is actually the light blue one is the highest. Then next, instead of being the same, we do find, as expected, higher neural synchronization in the proximal watching condition as well, showing that there is evidence for the role of touch and proximity in neural synchrony. But this was, of course, not enough. We also wanted to see if it's also related to different qualities of touch, and that's possible to do in the free play condition, in which we coded for affectionate touch specifically. And here we find that higher amounts of affectionate touch on the y-axis here were related to higher amounts of neural synchronization on the y-axis. And all these little points, so each point column is one dyad, so to say. And this positive relation supports our initial finding of neural synchronization during proximity, making it even more clearer in the sense that especially forms of affectionate touch, that those forms would be related to neural synchronization. So all in all, we find quite some evidence for the role of touch in neural synchronization in the current study. Neural synchrony occurs in physical proximity, not only when mother and infant are actively engaged with one another, but also when they're just merely watching a video together. Then we find even further evidence in the free play condition when the duration of affectionate touch is positively correlated with neural synchronization as well. And I feel like this gives us ample research ideas to take the research of neural synchronization and touch further in the developmental sample of, for example, how kangaroo care in preterm infants is related to neural synchronization between parent and child. However, there is still much data left in this current study, a very rich data set that we have, and we're going to look into further modalities and how they relate to neural synchronization, and such as affect, gaze, and vocalization, so keep an eye out for that. And we're also very fortunate to have measured attachment at 12 months in a majority of these infants, and we can take this data to look into how touch and how neural synchrony is related to potentially secure attachment at a later time point. I want to thank you very much for your attention, and I'm looking forward to your questions. If you can't be here, feel free to mail or DM me on Twitter. I'm happy to answer any questions. Thank you very much.
Hello to everyone.
Thank you, Trinh, for this amazing talk. I hope that you can turn on the camera. Wait a sec. I don't know. Okay, here you are. Hi, thank you for your talk, and I'm really looking forward for your upcoming results, especially those about the attachment style. Maybe I asked you for a spoiler at that time, but did you have a look to the cardiac synchrony? Because if I correctly understand, you also recorded the physiological or cardiac parameters. So do you have any data about the synchronization between the child and the mother in the different conditions that you test?
Yes, can you hear me? Yes, okay, all right. Yeah, we did have a look at the physiological synchrony, so the RSA synchrony in our study, and what we find is that they do show synchronization in the free play condition, but it wasn't related to proximity or to touch. So it's kind of similar to what we find for neural synchronization, but still different. And actually the behavioral correlate that was related to physiological synchronization was infant affect. So the more negative, or the longer the infant was in that negative affect state, the more physiological synchronization mother and infant showed. So it kind of hints towards this differentiation between neural paths and physiological paths, but I think this is something that we have to dive deeper into, and there's some converging evidence by another group in Aachen and at UEL London, but yeah, it's a very preliminary finding, I would say, at this point.
Yeah, I totally agree. Also, see what trigger what in a way. So if it's a sort of co-interaction and convergent interaction between the peripheral and the central synchrony, and also the different behaviors that you test, so the child to the mother or vice versa. That's of course we have a lot of model of that about that. Thank you so much. It's very interesting. Thank you for joining us and coming here and give your talk. So I think that